So for the first talk, it's a recorded talk because Dr. Eric Carlson is not being able to make it to Tokyo. I know a lot of people have been interested in this talk because it's called Game of Drones. So we'll be just playing the video now. Dr. Eric Carlson is Deputy Head of Virology Unit in Cambodia. So here we go.
Good morning, everyone. My name is Eric Carlson. I am the Deputy Head of Virology at the Institut Pasteur in Cambodia. I am so sorry that I could not join you there today. I was really planning on being there, but unfortunately I was unable to come. However, I am honored to be able to send you this recorded presentation where I'm going to tell you a little bit about our work on the development of field-forward environmental surveillance to advance early warning. So just a little bit about Cambodia, in case you all don't know. Cambodia is in the southern Indochina Peninsula. It's a least developed country with about an 18 million population. It is a hotspot of emerging and endemic infectious disease with a large dependence on agriculture and numerous interfaces where pathogens and people and animals all come and interact with complex hubs of biodiversity. We have been at this animal-human interface for over 25 years conducting surveillance in the virology unit of Institut Pasteur in Cambodia. And we're looking at a number of different diseases. I've highlighted some here, and I've bolted out influenza, because that's really what I'm going to use as an example today in this presentation. But you can see that we study a number of other things, including human diseases, animal-borne, and even unknown pathogens. So really what the problem here is that the question is really not if, but when the next pandemic will occur. And it's really only going to get worse unless you're better prepared, because the cost of an outbreak really goes up every second that we aren't. So what we focus on here, and my research focuses on here in Cambodia, is really early warning. So how are we going to get ahead of endemic and emerging diseases? And really that involves a number of different things, including anticipation, prevention, detection, and response. We need to be able to do all of these things at once to be able to prevent the next pandemic. Unfortunately, early warning at high-risk interfaces requires active surveillance. And active surveillance, for anyone that's done active surveillance, knows that it's really costly and time-consuming. And often it's not always done in the easiest of places. Here you see my team actually taking a boat out to a bird colony in one of the largest lakes here in Cambodia. So really what we've been focusing on is ways that we can improve surveillance for early warning. And what we focus on is two different things. One is sampling and one is testing. So in terms of sampling, we've really focused on environmental sampling. And they really provide a viable option to improve early warning. Environmental samples can speed up our detection. They are comprehensive, but also they improve biosafety of the individuals who are taking the samples. They improve animal welfare for the animals that are getting sampled. And because we can take less samples and get more information, they actually reduce cost, which can mean that we can do more sampling in a wider area of places. So there's a number of different places that we can do sampling within something like a live bird market that you see here in Phnom Penh. So we can do things like feathers, water, cages, surfaces. But what really we've been focused on the past few years is air, because we feel like it's much more likely that you're going to contract a virus in the air than it is from, say, kissing a chicken, right? So we've been doing a lot of validation studies in live bird markets. So here you see air being taken in a live bird market. This is a different kind of live bird market. And then of course in other interfaces. So this is actually a bat farm. What you see is a bunch of Scotophilus bats on the right that live in these palm fronds. And we've been taking different kinds of samples throughout the course of our surveillance in these places, including air. And we can take that while we're taking other samples from these interfaces. We can take it from bat roosts. So here you see my team sampling underneath a fruit bat roost inside of the pen itself. And then of course we can drag them into caves and do the same thing where we're taking air in the caves as well as taking samples at the same time. We also do this in other interfaces such as swine abattoirs. So we can use surfaces, species, swabs, but also air from inside these abattoirs to understand what's potentially going on in these interfaces as well. We can also now do this handheld. So we have these new handheld air samplers from AeroCollect. Here you see us taking samples from some ducks, but we can also really target potential aerosol generating areas such as this defeathering machine here. These are really nice because we can actually target individual animals within the population or very small areas within a larger area while we're taking concurrent larger samples with our larger sampling machines. These really provide us a way of sampling not only large populations like you see here with about 1 million bats flying out of that cave and that air sampler below it, but also really targeting things while we're doing other kinds of sampling. So here on the right, you see us taking air samples from individual bats while we're collecting samples from them in the forest itself. We can use these to our advantage in places that we may not be able to sample very easily, in things like the wildlife trade as well as captive animals. In case anyone wants to know how you sample a civet, here's how it's done. You offer it some banana and then you stick a swab down its throat. Here we're also taking air samples at the same time. And we can even use these remotely. They only weigh about a pack of cards. So we've done things like strap them to drones and then strap them to what I call terrestrial drones. So these are really just $60 remote control cars that we can then drive into places or fly into places that we may not have good access to, or we may not be allowed to actually walk around and go into. And we have done this in the wild itself. So we fly drones into bat caves and things like that to collect samples from places that we may not be able to reach. And we use these terrestrial drones in places like swine abattoirs and things like that to collect samples for influenza and African swine fever testing. We also collect a number of other types of samples, so things like classical rope swabs, water, etc., which allow us to really get an idea of what's going on in that environment. So does it work? And which is really a big question that I get all the time. And the answer is yes, it does work, and it works in a gradient style manner. So if we look in chickens or ducks within a market, we do find avian influenza virus, but we can find it in a gradient pattern between slaughter, storage, and food sales, which is really outside of these areas, in a gradient pattern as well with reducing viral load. While we don't always detect it in the air, we are detecting it about 50% of the time in our samples, which does correlate with what we detect in the birds itself. So the other thing that I wanted to talk about today, and I'm sorry that I can't go into more, but the other thing I really want to talk about was improvements in testing. So how can we expand our capacity for faster diagnostics and understanding? So we've been doing a lot of different things on improving testing and diagnostics for early warning, but of course we want to ensure both sensitivity and specificity, but also increase the speed and versatility of our diagnostics, and of course be able to discover new high-risk pathogens or subtypes. So one of the things really we're working with is nanopore-based sequencing. We have been working together with our colleagues at Johns Hopkins Applied Physics Laboratory to really advance avian influenza sequencing using integrated indexing and field-forward bioinformatics. So we've been using the unique primers coupled with integrated nanopore barcodes, which allow us to drop the sequencing time that we have for influenza from 33 hours on Illumina down to about 7 hours in our hands in the lab from sample to full genome sequence. What this does is it allows us to couple it with real-time bioinformatics, so like you see here, where the samples are actually being investigated in real time by the data coming off the nanopore machines and actually being subtyped within 5 minutes of the first set of runs actually coming off the machine. That really increases our ability to detect and respond to things. So we've used this not only for surveillance, where we're looking for prevalence, introduction, and risks of major subtypes of concern, but we've also used it for understudied subtypes, things like H6, H10, H14, and also for responding to cases of human spillover. So, we've used this successfully to go from sample to sequence in very, very short amount of time, which are critical for response here in Cambodia for the government here and other individuals to actually respond. So we've responded to 2 cases of H9 in young children, one in 2021 and one in 2022. And then, of course, a number of H5 infections between early 2023 and early this year, where we were actually able to do genomic epidemiology of these cases between both the birds and the humans to understand how they were actually circulating within the country, and understanding that they are 2321C viruses, similar to what's been circulating here for 10 years, and not necessarily 2344B viruses, which are circulating globally. And another thing that we're doing is actually looking at pan-family viral surveillance. So this is really building off expertise that was built during the PREDICT 1 and PREDICT 2 programs, but again, incorporating these integrated barcodes. We've made highly multiplex systems that take 1 round of 2-hour PCR, where we can detect multiple viral families. So here you see we're detecting multiple types of influenza, as well as beta coronaviruses and flaviviruses and even rabies, all within the same assay here. And then these can also be done in real time because they are indexed barcodes. So each individual sample actually gets an indexed barcode, as you see. They are barcoding in real time. We can then input that into our real-time bioinformatics system, and those then become actually subtyped in real time, even down to the subtype of influenza, and actually allow us to quickly assess what's in our samples within 5 hours in a field forward manner. So you can see how quickly we can use these things very, very well. The other thing that we're doing is looking at if we can combine environmental surveillance with metagenomics for even more rapid information. So here we're taking air samples in a live bird market. So combining metagenomics environmental samples does a lot of different things. It gives us unbiased and broad ability to look at samples. It's dynamic, we can get functional information, and we can even do pathogen discovery. What you can see here is that when we run metagenomics on, say, a poultry cloacal swab, the majority of that is going to be influenza, which is what we expect because that bird is infected with influenza. But if we just look at a single air sample from that same time period, we're still getting that influenza virus. In fact, we're getting the whole genome of that influenza virus, but we're getting a number of other diseases in that sample that we actually can look at and understand the high-risk interface that we're looking at. A lot of people ask me how that compares with real-time PCR, which is the gold standard. And you'll see that sequencing actually gives us more detections of these viruses compared to PCR in and of itself. So that really lends itself to the question of, is NGS more sensitive than PCR? And that really depends on how you define your cutoff values and what's positive. But here you can clearly see from a spike-in experiment that we may not be detecting something over that's over the actual positivity rate for PCR, but we're still getting a good amount of that genome at over 100x within that sequencing run. So, it really is something that we're investigating further now. And so what you can really see if we look at all these things together, because we've run hundreds of samples through these systems, is that air really gives us a really nice diversity of what's going on. And it does actually reflect, and I don't have time to go into this too much today, it actually reflects exactly what's in the birds down to a contig level, plus the number of other viruses which are present within that system. So in the future, I would love to talk to you more. You all are welcome. You can come to Cambodia and work with us and do things here in the field. But in the future, we're really planning on scaling up to expand our surveillance to markets all over Southeast Asia. We're going to continue to refine novel techniques. We've a number of interesting things in the pipeline overall, including novel data visualization for reporting and action. Excuse me. We're going to refine our in-field testing algorithm. So really being able to do this in the field. And this year we are actually going to be doing this in the field in real time and showing that we can actually use environmental surveillance with real-time in-field sequencing and metagenomics to expand our surveillance capacity for early warning. And of course, the future is now. I think you've probably seen some information how wash water and wastewater from dairy farms and in other places are actually being used to detect H5. They are taking environmental samples from dairy farms. And these are absolutely critical things that we can use when potentially they don't want individuals on these high-risk areas or in commercial places, or to really get an idea of what's going on in the community overall. And with that, I'll say thank you. My time's running out a bit, but I can be contacted in a number of different things here. My email, as well as several social media platforms. And then, of course, I have to thank all of the people we work with. I have to thank Bill and Melinda Gates, especially for funding the majority of this work, as well as work under the CREED program and an IAEA Zodiac project. And then, of course, everyone that we work with overall. Please feel free to reach out with any questions. Again, I'm happy to host people here in Cambodia, and I do hope that I will be able to join you next time.
Dr. Carlson, I don't want to be here from Cambodia, but there's a current avian flu outbreak there, which currently is not yet human-to-human transmission that he's observing, but is running their diagnostics lab in the country and had to stay behind. So I don't know if he's on Zoom, but someone we collaborated with the Pasteur Network, which I know is also part of the Working Two Trends Lab. So I think since he doesn't seem to be on, we'll just move on. But he sends his regrets. He had a ticket in a hotel room and a visa and everything, but something that said people are getting weird viruses and we need your help. So if anyone has things you need on drones or caves, he's certainly the guy to go to. Also, he gets about one new tattoo every 6 months. So if anyone likes to get tattoos, he's also your guy. But, okay, thanks. The intro.
So up next, we'll have our keynote speaker, Dr. Nikos Kripidis. He's a senior scientist in the DOE Joint Genome Institute. He's also the head of metagenomics program and has led the combined microbial genome and metagenome program since 2011. His work focuses in microbiome, bioinformatics, big data, and data science.
Konnichiwa. That exhausts my Japanese. I would like to thank Grace for the second year of invitation at MetaSUB. I was for the first time I was in Florida in the first meeting. Unfortunately, this was next to the pitch, and I missed most of the meeting. I have to admit, but this one I'm really enjoying so far. So the main focus of the talk today will be about the global virus, but I would like to give a more general. I would like to start with a more general overview of the directions we're taking at the JGI in the prokaryote super program and the microbiome data science. Was there something wrong?
Sorry, come on, everyone.
I can turn from here.
One moment, everyone, so sorry.
Oh, yeah, that works. Okay, great. Thank you. All right. And I would like to start with a very brief overview of where is the place I'm working at, which is the Joint Genome Institute at Berkeley National Lab in Berkeley, California. So this is the U.S. Department of Energy sequencing facility, science facility, actually. We're doing a lot of sequencing, as you can see last year, more than 700 databases of sequences. We're doing DNA synthesis as well, and there is metabolomics lab with a lot of metabolomics analysis running every year. And there's a really big part which is data analysis. The mission is, as part of the US Energy User Facility, we provide advanced genomic capabilities to pretty much to address major DOE mission areas. And you can see here, these are pretty much bioenergy, carbon cycling, biochemistry. Actually, it's pretty broad. Pretty much everything except human-related data analysis or sequencing and data analysis. There are 6 major science programs, 2 in eukaryotes, plants and fungi, 2 in prokaryotes, plants, metagenomes and microbes, and then there is the DNA synthesis and metabolomics. And below you can see the technology and products, a large number both for DNA, advanced genomic capabilities, computational analysis, and DNA synthesis. I'm leading the Microbiome Data Science Trust for the more than the last 10 years, and which pretty much covers different dimensions of diversity, and that includes microbial, functional, and viral diversity. And the focus today will be more, I will present it, about half of the talk will be about projects we've already published, but I will, most of the second half of the talk will be about unpublished data and new directions we're taking, and where we're going with in all of these 3 directions. So why I would like for this is for my friend Venkat, who keeps on saying data is not as important. So data, I think most of us know that we are living in a data world and data is power. And why is that? The main reason is, or one of the reasons, in my view, it's one of the most important reasons, is because data is what economists are calling non-rival. What does this mean? Nearly all economic goods and services are rival. That means that if a product is used from somebody, it cannot be used at the same time or ever again by somebody else. Data is exactly the opposite. They are non-rival. They can be used simultaneously, repeatedly, by a number of firms or people without being diminished. So data can be used and reused, and this is part of their power and why they have become, why we're saying today data is power. And of course, it's extremely important to always remember that the more freely the data flows, the more likely it is to spark new ideas. And this is important, particularly at the time that a lot of scientists want to keep their data private or hold them for a long period of time, rather than sharing with everybody else. And most of the talk will also will emphasize the importance of analyzing and reanalyzing the data, and I hope I will be able to convince you that none of what we do is endangering the work that other people are doing. And that probably is the punchline of the entire talk. So the system we're using for doing all the data analysis, data integration and data analysis, the JHI is called ING, standing for Integrated Microbial Genomes. And this integrates a large number of different data types, genomes, functions, genes, metadata, clusters, transcriptomics data like transcriptomics, proteomics. In the last few years, we have integrated a large number. We're identifying viruses and plasmids and integrating them into the system. There are currently more than 200,000 datasets, and altogether, isolate genomes and metagenomes, we have more than 80 billion genes. And this is an enormous database. There are no 80 billion genes available at NCBI, for example. And we'll come in more detail, we'll discuss in more detail viruses and plasmids later on. So a couple of science vignettes in each of the microbial and functional diversity, and then I'll move to the viral diversity. So we're at the point that although most of the microbial genomics through the last 30 years has been actually sequencing of isolate genomes, the last 5 years, the whole field of metagenomics and the reconstruction of genomes and metagenomes has exploded. And we're actually at the point of having hundreds of thousands of genomes that are not coming from isolated genomes, but from the environment. And this is gonna explode in the next few years as well, as we keep on sequencing more metagenomes. Some of the major areas we have been working in this direction is the human gut and overall all metagenomic data regarding JTI, what we call GEM, the genomic catalog of Earth microbiomes, trying to reconstruct massive number of marks. And in both of those cases, we reconstructed several tens of thousands of genomes. And of course, there are a lot of other efforts from the community producing thousands, hundreds of tens and hundreds of thousands of marks, medium quality and high quality. There are a lot of applications. Why is this important? This is changing the field in several different ways. It is providing, marks are providing new reference genomes for metagenomic analysis, and particularly for the vast uncultured diversity. New ideas to culture, uncultivated organs, of course, and new diagnostics for disease. But as always, when you go into new territories, new, completely unexpected or new ideas or new observations come. And that's what I'm gonna present in the next few slides. One of what is, for me at least, one of the most exciting observations we have done in the last few years by collecting all this data. And this is still unpublished. So we have the question of how many genomes from a cultured organism can be recovered from metagenomes. In order to address that question, what we did is we took more than 1 million, 1.4 million actually, genomes from isolated organisms, from GenBank. And we collected also all the marks we had in ING, and also we went there one step further and we collected all the marks from NCBI as well. And then what we asked is how big is the overlap? So what we used is the ANI, the average nucleotide identity, in order to group those two groups. And we ended up identifying species, and that's what the ANI pretty much giving you, what you use that were exclusively consisting of isolates and what you use exclusively consisting of marks, as you can see in those two cases. So the important question is how big was the overlap? Any guess, anybody? How big will be the overlap? How many marks will include, how many of the use of marks will include isolates? The number was astonishingly small, 5%. And that begs the question why we cannot recover isolates from any environment. So if we think for a minute what marks are, they are genomes reconstructed, the genomes reconstructed from the genomes represent the most abundant organs in that environment, right? That's why we can capture them. We have enough coverage that we can do assembly. So that immediately means that most of the isolated organisms are not abundant in any environment. So what would that mean? That is one observation. The second observation, and we have to couple that with the second observation that I don't have here, when we looked in more detail what the marks are, the vast majority of them have metabolic deficiencies. That's why they remain uncultured. They cannot be isolated because they do have metabolic deficiencies. And because of that, they do rely on a partner from that particular environment. So the marks have metabolic deficiencies and can then at the same time we observe that they are abundant in the environment, in most environments. So what does this mean? If you want to dominate an environment, you have to work with others. You have to create this biotic relationship, while if you don't need anybody else, you go at the bottom of the food chain pretty much and you will be extremely rare. So you become rare biosphere. So this is the hypothesis we have and we're trying to explore further by doing further analysis into that. And at the same time, try to understand what is the diversity of metabolic complementarity and what are the pathways that are exchanged between all environments across the whole line. So looking into this in more detail, we have the second question, which is how much diversity is actually then not captured from marks. And how can we address that question? We have been trying to address this question pretty much for more than 10 years with 16S. 16S though is not the ideal tool when you're looking into marks. And the reason is that most marks don't have 16S for several reasons. Contents that carry 16S don't ping with the rest of the mark, even the high quality marks. Most of the high quality marks don't have 16S. So we needed a different tool, and what we used is combination of 5 single copy marker genes from different varying size, from let's say 300 amino acids all the way up to 1,000 amino acids. 300 amino acids you can assemble much better, genes with 300 amino acids, 1,000 much less. But still, that gave us the whole, when we cluster all those into A and I groups, or OTUs, we got the picture you are seeing here, which is the gray is pretty much all the diversity. This is species. The OTU diversity that is not captured by major isolates. So this is what we believe is an indication of the diversity of the rare biosphere. And why do I call it rare biosphere? Because we do not capture it from any of the 50,000 metagenomes that we have so far. At least we have analyzed in ING. So the question then is, how can we tap into this diversity of 125 or 130 or probably more? This is only what we can see based again on the single copy marker genes of the assembled genes. Definitely the diversity is much bigger, but we can definitely say that there are 100 to 130,000 OTUs that have not been captured by much so far. And the main reason again is most likely they are very low abundance. They have very low abundance, so we cannot capture them. Venkat may have suggested or may have come up with an approach to actually go into this by implementing the NASA cleanroom approach, which is try to get rid of the dominant organisms in any environment, which are masking the lower diversity or the rare biosphere, and then give them a chance to maybe grow on a plate. So that's a hypothesis we have been discussing with Venkat, and we'll try to follow. And here we see we use DDP3 in order to organize the diversity into the different, the 5 phylogenetic taxonomic levels, and we see. Oops, I just go here. We see here the gray is possible novel phyla, so we're trying to go after those and see if we can capture them through marks. Functional diversity, the main emphasis there over the years has been to find novel versions of biotechnology, of genes with important biotechnological applications. For example, new recombinases, or new Cas genes, or new CRISPR systems, and so forth. So we had a long, several-year collaboration with Jennifer Doudna's lab, where she was looking, where we were doing the mining across all the data to find novel Cas genes, or CRISPR genes, Cas9 genes, or other novel Cas genes, and new CRISPR systems. So this is one approach, pretty much, and that's a major area worldwide, using metagenomic data to find new versions of already known functions, right? So, but all of these functions rely on the functionality or the diversity of the functions that we know from isolated organisms. What about completely novel functions that we haven't seen before from isolated organisms? So this was the question we tried to address, and we call that functional dark matter. And the main question was, how large is the unknown functional diversity out there? So what we did, this is where the work that George Pavlopoulos did, was published last year, less than a year, in Nature. So what he did is he took all isolated genomes, and he collected all the genes from the isolated organisms, and he clustered them into clusters of 100, at least 100 proteins or more, 100 genes or more, and he ended up finding, here, he ended up finding around 92,000 clusters. He did exactly the same with metagenomic genes. After he filtered all the metagenome genes, he started with 8 billion genes. He filtered any genes that had hit to isolates or PFAM, or even remote similarity to genes from isolated organisms, and he ended up with 1.2 billion genes. He clustered those and he got 113,000 clusters. So more than double the functional diversity that we previously had. Of course, these were all hypothetical protein families. What can you do with those? There is no way to predict the function. So what we did was habitat distribution. So we identified pretty much functions that were found only let's say, on the human body, or on soil, or marine, and so forth. And then we used AlphaFold to cluster on the clusters in order to predict structural similarity. And we're able to predict structure on a small percentage of those, 30% overall, with a few completely novel folds as well. So this was done, this effort started several years ago. We had a lot of technical problems until it was published last year. Now we're at a point that we can repeat it extremely fast, and we have already rerun it on the new data we have in Nigeria. We have more than tripled the number of protein families we have here. So that's in preparation for publication. In addition to the paper, we created a database that people can actually access all these novel protein families and mine, look at the structures, use the sequences for further analysis. And this was published in the NER special issue, and I was very happy to see that this was identified as the breakthrough article of the NER special database issue this year. And we're getting into viral diversity. So by viruses, first of all, the size, the number of viruses out there. So there are orders of magnitudes more viruses. You already know there are more bacteria on Earth than there are stars in the universe, right? Well, there are more viruses out there than there are way more viruses than there are bacteria. And this the image on the left is taken from a paper that was published by Furman's lab, and actually shows here the lighter points are bacteria, and the dots, the smaller dots are viruses. So this is a real image. So, and they estimated how far would you go if you take all phages on Earth and you connect them next to each other. How far will you go? Can you guess, anybody? Will you go from here to Europe or to California?
Alpha Centauri.
Sorry?
Alpha Centauri.
Well, the estimate is more than 2 times the diameter of the Milky Way. So that's very good prediction, very good estimate. So there are a lot of reasons why viruses, and we know they control pretty much bacteria, and a lot of through that, a lot of biochemical and ecological dimensions, let's say, on the planet. But nevertheless, we were not able to start them until a few years ago. And the major difference, let's say, with bacteria is we don't, and we still don't have a common one universal gene, like we have 16S, for example, or other single copy marker genes we have for bacteria and archaea. And we didn't have any cultures. Again, like at least what we have for bacteria and how we started microbial genomics through all the cultures we have. Nevertheless, through the last, decade, we started getting new approaches in order to study those. The traditional ones were through isolates, through microbial isolates, identify the phages, or through single viral genomes as well. The approaches that have been developed mostly the last 10 years is viral metagenomics and microbial metagenomics. So let's say take whole metagenomes and try to find viruses. And each one of those have different advantages and disadvantages. The interesting thing here is that the overlap between when you take a sample and you do WGS, you sequence the whole sample, or you try to do the viral component only, the overlap is very little, probably less than 20 or 30%. And that shows also that both, ideally, although it's more expensive, but ideally you should use both approaches, both whole genome and if you want to zoom in and identify less abundant, let's say, viruses as well, or different types of viruses, you have to do viral amplification as well. So definitely the recommendation is, if you're interested in that part, do both, to follow both approaches. And as I said, until recently, until less than 10 years ago, most of the viruses we had were from isolated organisms. And there was a big change here in around 2016. So that was the first time we had a really big, large-scale analysis of discovery of uncultured viruses. And most of that was coming from a large-scale analysis we did at the GTI. So that's what we thought at that time is why don't we take all available metagenomes from cats? And that was a very small number at the time, 4,300 samples. These were already sequenced, analyzed for bacteria, etc., and they were just sitting there. And we thought, how about if we'll try to identify viruses? So we came up with a new pipeline after a huge amount of manual work because we had to create a manually created list of 24,000 HMMs that were viral specific. That's the other problem. You cannot just take all viral genes because there is a lot of integration of bacterial genes or archaeal genes there. So you have to make sure that you're using only true, let's say, phase genes or viral genes. So after we did that, we threw the bait, let's say, to the all metagenomic data, and we ended up identifying with one effort, 125,000 viral genomes. At the time that we had only 3,000 available genomes in general. This really gave us, opened a new window, it gave us a new approach or a new tool to go across different samples, but also try to identify, zoom in different types of viruses, for example, giant viruses, or viruses, or virophages, which are actually infecting viruses. And one of the last big efforts we had was on the human cat, as you can see here on the right as well, by zooming into the human cat as well. That's the only slide I have from this study. So with, again, with a single effort, we were able to identify almost 200,000 different viral genomes. And what I want you to show here is that 65% of the phylogenetic diversity of all the viral genomes we had at that time were coming from this study. So it dramatically increased, and here you can see on the tree, this is actually a tree from Cotovirales, and all the green are novel lineages that we didn't have before. And the rest is, here is taxonomy, host taxonomy, and then viral taxonomy as well, and they increase from this study. At the same time, we're working with Chris on the MetaSUB, big MetaSUB, the first large-scale analysis that you all know, and then we did the viral identification, which led to identification of a little over 16,000 different viral condigs from these samples, and with a large part of that was completely novel diversity. Novel groups were representing more than 90% actually. At the time that we were working on the MetaSUB, we started working also on metatranscriptomics data. We had a JTI, and we initiated a collaboration with NCBI, the Eugene Kunin's group at NCBI, and with a group at Tel Aviv. And extremely talented PhD student there, Yuri Neri, who's actually postdoc right now at JTI. And what we did is we took all of the available metatranscriptome samples we had at that time, which was around a little over 5,000 samples, and we used the RDRP-based approach to identify condigs that had the RDRP gene. So that was a pretty complicated overall pipeline, but the punchline here, what I want to focus was that we ended up with identification of 2.6 million viral condigs, which were RNA viruses, which when we grouped were around 124,000 different classes representing different species. And we identified a couple of, two actually, putative novel phyla, RNA viral phyla, and several different families as well. And there was a big diversity, habitat diversity, based on the availability of samples we had at that time, and we also saw that we're not really reaching a plateau here or in any habitat overall, here or in any particular habitat. So what about computational tools? So most of this work was done either with a single gene like the RDRP gene or with the 24,000 manually curated HMMs we had. And these were good enough to get us to the first stage, but we knew that we could do much better. And the available tools that were out there were not also available, were not also able to capture the overall viral diversity that we could see, at least manually. This was one problem, and the second problem is what was identified in a publication, in a community publication, standards publication a few years ago, which was identification of genome quality. So we don't know, we didn't have something equivalent to check-in which is for bacteria. We didn't have something equivalent for viruses. So we didn't know the level of completion and the level of contamination of a viral, of what we identified as a viral content. Stephen Neifert, who was a scientist at JTI at the time, undertook this task, and he came up with a tool that we call CheckV, analogous to check-in, and this is a tool that is assessing the quality and completeness of metagenome sample viral genomes. So it follows several different steps. The first step is remove host contamination, and it's doing that by a very large database of viral and bacterial genes, so it can identify the borders between, if it's a prophase and integrated, we can identify where exactly the borders are, of course, with a small percentage of error, and then identification of estimate of the genome completion. And this is based on conversion with not only with a known isolated phases, but also with a curated database of circular viral genomes that we predict or we believe are complete. And the next step is prediction of those genomes, so identification, and that is done using direct terminal repeats from the context, and there is some false positives there that we're trying to work on right now and improve. And the final output is percentage of completion and possible contamination for all the phase genomes. So this is the only tool that is out there actually, so it's widely used in the community. There is no other tool that is doing what CheckV is providing. So it has become a standard in the field. And then we, as I said, we felt that we still do not meet, cannot meet all of the, none of the tools we have can give us the diversity, viral diversity that we believe is out there. So this was another challenge that Antonio Camargo, another scientist at the lab, tried, and he came up with a tool that he called, he named Genomad, based on Nomad, and Genome and Nomad, so pretty much jumping or moving around, and the tool is not only predicting viruses, but also plasmids. So this is, there are a few additional tools that are doing the same, which is predicting both plasmids and viruses. But this is the only tool that's actually using a combination of sequence-based approaches and sequence-independent neural network, pretty much, or machine learning approaches to identify, to predict, to classify viruses into chromosomes, plasmids, or viruses. And the final output is actually pretty rich. It gives not only identification of contigs as viral or plasmids, but also taxonomy, genetic code if it's different, functional annotation, hallmark genes, and also some additional functional information like conjugation genes and ARPs and so forth. As I said, it's doing that with a combination of HMM, sequence-based, and sequence-dependent and independent. The sequence-dependent is based now on a combination of 227,000 microgenes. That's an order of magnitude more compared to the first pipeline we had. And any contig is classified as either chromosome or viral or plasmid, and there is a score associated with that. So you can select how confident you want to how confident assignments you want to have when you run the tool. This is benchmarking. I'm sure you cannot read back there, but I just wanted to point out that there is detailed benchmarking done with all other available tools. It's outperforming all other tools, both for viruses and plasmids, and it's working even much better actually in the lower contig size, which is more challenging as well, but also performs very well in local context. There was a great performance with endoscopics as well, because as I mentioned, the first paper we had was only on the RDRP base. but we saw a lot of condigs had RNA viral genes and no RDRP. And these were, of course, fragments of condigs that did not include, or it could be segmented viruses. So what we're trying to show here is that when this is RDRP based, and when we used the tool, we also found a large number of condigs that did not have RDRP, and some of those were actually binding with the RDRP condigs. There was a great performance on diversity as well, and we're still not reaching a plateau. This is the diversity across the globe of the RNA viruses we found using genomads and of the actual DNA and RNA viruses and the phylogenetic diversity here. We have organized all of this data into a database, a specialized database that we call INGPR, and we update this every 2 years, and we have also the CheckV values. So this is 15 right now. I'm going to come back to that in a minute. We have 15 million, more than 15 million viral condigs already. So we launched the database back in 2016 with the first study I presented, and then updating every 2 years. So blue here is the number of condigs, orange is the number of OTUs. And this is where we went when we applied genomads into all the genomic data we have into ING. So we have a little over 2 million viral OTUs right now, and more than 15 million viral condigs. And this keeps on constantly getting updated. But as I said, Genomad can also predict plasmids, and by applying it across all isolate genomes and metagenomes in ING, we're able to find almost 700,000 plasmids. These are the high confidence. There is a much bigger list of several million that they seem to be plasmids, but we didn't include in the database because it requires more detailed checking, let's say. But there is a large number of plasmids from any habitat here, from any environment, including soil. And we're actually looking into soil more in more detail, and we're preparing publication study for that. But all these data are available for downloads. You can go either viruses or plasmids, download and use them and do any analysis you like. So what's next? How much time do we have left?
Around 20 minutes.
Okay, great. This comes, I think, the more exciting part here, which is what's next and what has not been published. And what I would like to briefly present is 2 studies. One is a unified catalog of the human gut, and the other is a completely novel, I believe, direction, which is what we call a viral infection approach. So the first one, which is in preparation, and again, an effort led by Antonio. As I mentioned before, we published a few years ago the human gut virome database with 180,000 viral condigs. In less than a year after we published it, there were several publications coming up with completely new lists from different studies, new lists of human gut viruses. So what we decided to do then is collect all of these studies, and combine them in what we call a unified human gut virome catalog, which was actually led at that time by Stephen. So we used all the tools we have developed, and this is the geographic distribution of all the combined databases. 77% of those fortunately had geospatial data, so we can point them in exact positioning across the globe. And when we combine all these different databases, we ended up having a database of creating almost 100,000 viral OTUs. And this is the distribution here in terms of CheckV, completes. About 60% is complete, or more than 90% complete, and 40% medium, what we call medium completeness, 50 to 90%. And then here on the right, on the top, you can see that around 44, less than half of those were on the first database we built, MGP. And all other databases had also much less diversity overall. At the same time, we realized we need to classify them into not just OTUs, but to higher level, let's say general and families, and we came up with an approach based on a combination of DNA and protein identity, and we're able to classify all of this in the 100,000 viral OTUs into 20,000 genera and 500 common families, most of which are actually were not previously known. We see that we're not reaching a plateau in the VOTU accumulation, but we have been reaching a plateau in the family. So overall, bigger diversity, I think we're coming up to a plateau or complete, let's say, diversity but we're far from seeing something like that, at least for the beauty use. We also we tried to organize all the protein sequences into families and try to improve the functional annotation there. So we started with 37 million genes from all those, from the 100,000 BTUs. We did clustering, went up with 1 million clusters, and then we also applied AlphaFold and structure prediction pretty much. A very small number of the protein, the 1 million clusters did have a structure prediction, only 56,000 out of the 1 million. But the most interesting part here, or very interesting part, is that these clusters, these 50,000 clusters, the 5% of all the clusters represented 76% of all the protein sequence, all the protein sequence diversity. And this is because these are the largest clusters out there. So whatever remained, the remaining, let's say, 950,000 clusters had the remaining 25% of the sequences. And what we observed here as well was that although we have reached the diversity of the structural diversity, we have reached a plateau a long time ago, and we're reaching a plateau as well in the non-singleton protein families. We're not yet reaching a plateau in the singletons as well. So as we keep on adding more sequences, we are seeing more proteins as well, but singletons. When we compare the number of protein structures with functional annotations, we've seen that 50%, about half of the structures here, don't have annotations in any of the databases, let's say PFAM, COC, etc. So we now have structural predictions, but no functional annotations for about 50% of the clusters. And this, I just want to mention here, to show here that the overall functional annotation of all those proteins improves significantly when we're adding the structural prediction component as well. And coming to the last part, to the last vignette, which is the viral infection networks, which is actually currently in review. This was an effort led by a postdoc in the lab, Rayon Freeman. So the idea was the following. I think everybody is familiar with what the CRISPR system is, and we have been using that spacer viral sequence connection in order to predict the host of the virus. So if there is a sequence similarity identity between a fade sequence and a spacer, we are predicting that this virus has this conjugate, let's say, this host, this conjugate as the host, this genome as the host. So most of the publications that are out there, actually I would say exclusively, and not just most, but every publication out there that has been doing that has been using CRISPR systems from isolate genomes. And there is a good reason for that, and the reason is you need to know what is the taxonomy of what is the origin of this organism, which is the host. If you take a conjugate from a metagenome that has absolutely no taxonomic information, what are you going to do with that? But this is exactly what we decided to go for, again, for a very specific reason that I'm going to explain in a minute. So we ignored all of the conjugates with CRISPR systems, with CRISPR sequences from isolates, and we went to the metagenomes. So we used a previous version of INGBR before we went to the 15 million, we had 2.3 million. This is INGBR version 3, which was around 900,000 VOTUs. And we got all the CRISPR arrays from the metagenomes we had in ING, around 10 million, more than 60 million spacers. And we did the conversion. This took 3 months, more than 3 months actually to complete. And what was the goal? What did we want to achieve? We wanted to identify cases like that, that is conjugates that connect, arrays that connect multiple viruses, or at least 2 or more. And the connection can be either a single virus with a spacer, or it could be more than one virus with the same spacer. And this could be actually the level of different VOTUs, not just different sequences, because this could be all part of the same. If all of these viruses were part of the same VOTU, we threw away, we didn't care. We were looking for distinct viral specific connections to the same array. What does it mean to the same array? The same host. So even if we don't know what is the identity of the host, right? The conjugate is pretty short, and we cannot say what organism it is. We can still say that all of these viruses are infecting the same host. And it can be a single array, or it can be multiple arrays, as long as they're on the same conjugate. because a condict could be carrying multiple different arrays. So when we did that, so this is pretty much our definition of viral infection networks. Viruses, two or more viruses connecting to the same array. So the goal here was pretty much to identify co-infecting viruses. We cannot say co-infecting because we cannot predict that they were infecting the cell at the same time, right? But they were definitely infecting the same host, through different periods. So this is the approach that Brajon took in order to create those networks. He started with all of the IMGDR viruses, IMGDR version 3. Oops. Okay. He used all the spaces as well, and by doing a conversion, he tried to identify that. When we did that, we ended up with a huge list, which was 5 million spacers, connected to 2.3 million viruses, actually almost all of the viruses. Then we realized that we have to do a lot of filtering, which I'm not going to go into a lot of detail, but after applying very stringent thresholds, we ended up with 50,000 OTUs connecting to 500,000 arrays and 2 million spaces. So this was our final data set. So what did we do with that? We soon realized after looking at the data that we can have two different types of bins. One is what we call array-centric bins, which is what we see here. So multiple viruses, this was the original idea, multiple different VOTUs connecting to the same array, right? So you can see here these three or these two. So in this case, we have two different array-centric bins. Array bin 1, which has this network, and array bin 2, which has this network. And you can imagine that you can end up having hundreds of viruses connected to the same array. Overall, we identify a little over 500,000 VOTU array-centric bins. This was one of, and then we can organize all of these connections. The minimum, as I said, is 2, and then what was the largest? The largest was 1,100 different VOTUs connected to a single array. So this was the array, and you can see the white is spaces that don't hit anything, any viruses. So the vast majority of spaces hit something, and most of those are hitting actually multiple different VOTUs, with a total of 1,100 distinct VOTUs connected to this array. And this is from human gut actually, and fecal material. So this array is connecting 8,600 viral condigs, organized into 1,100 VOTUs. So that was the record here. But there are several other with 700, 600, 500, and so forth. The second type of network is what we call VOTU-centric. So in the same picture here, instead of looking this as a single array, we can ask with how many other viruses V1 is found. And V1, which is here, can be found, is connecting to all of these other condigs, not only viruses, not only from this array, but also from this array. So we're looking across all arrays with how many other viruses, any VOTU can connect. And this becomes the VOTU-centric bins. And in this case, we have 4 different distinct bins. So what can we do with that? And actually, this was the first version of the reviews we got. This is an interesting idea, but what can you do with that? We still don't know. we think that this is a very, very exciting and very interesting, different way of looking at all the viral networks, but we have only 2 applications so far, and we're trying to come up with additional ideas of exploring all this. And we got 50,000. This is the number of VOTU-centric bins here is as large as the number of VOTUs in the networks, which is 50,000. And again, this can grow dramatically because, first of all, we used only 2.3 million viruses to begin with, which was the previous version. We didn't use the 15 million. And second, we did enormous filtering in order to come down to a smaller number that we can handle easier as the first approach. In this case, this is the largest VOTU-centric bin. Again, we see the same picture. The virus that's connected to the largest number of other viruses is this one, which is connecting to 4,900 other VOTUs, which is this one. This virus is connecting 276 different arrays with this one, in 45 arrays with this one, in 121 arrays with this one, and so forth. So this can give us, we can very easily now get co-occurrences, let's say viruses that are co-occurring with another virus. So the two approaches or the two applications we got are the following. First of all, when we saw that through the concurrence, when we saw that two viruses are co-occurring in several different arrays, we thought that this could represent possible fragments of the same virus. And then we thought, rather than trying to do the assembly, just compare with IMCPR version 4, which was much more improved, let's say, version. So indeed, when we did that, we saw for at least 3 viruses. The green and the orange here are 2 different fragments, 2 different viruses connecting to this array. In IMCPR4 is a single conjugate. So we just captured 2 different viruses, and that's why they're different VLTUs, because there's no overlap. These are different conjugates, these different fragments of the same virus. So we can actually use that now to identify fragments or candidate fragments of the same virus, and then trying to come up with additional ways of doing that large scale. The second one is improving host assignment. So again, this is a very complicated slide to show, but what I'm trying to point out here with the blue versus green, green are all the new assignments, host assignments of viruses that, for which we didn't know the host before, across all these different hosts. And how do we do that? When we have 2 viruses again connecting, constantly found 2 viruses, 2 different VLTUs, from which we know the host of one, but we don't know the host of the other. If the host of the first one has a consistent, is consistent, and we don't see a big diversity of different hosts, then we can actually, we believe we can accurately predict the host for the second as well. And by applying that, we were able to increase the host assignment to more than 30% additional viral ODUs that we didn't have, for which we didn't have a host before. And again, this case as we add more data. So the importance of all that, the whole field of phage therapy is in a way rejuvenated with the enormous addition of all these phages out there. So there is a large number of startup companies in the field, and for a number of different applications. First of all, they're used, a lot of companies are trying to come up with a phage cocktail to kill bacteria. Identify phage enzymes, actually we've been doing that from the human gut as well. Phage antibiotic synergies is coming up again majorly in the field, in the overall field. Engineer phages to do different things, not only necessarily kill, but maybe assist bacteria as well. And the overall phage and CRISPR interaction, let's say, in warfare is extremely exciting area. And these are some of the, again, some of the areas: pest control, food industry, combating global warming for cow-rumen bacteria, and decreasing antibiotic resistance as well. So these are overall areas where phage sequences can play a major role. I was looking at the internet to find what are the projections for the overall monetary value. Where is the global value of the phage? And I was very disappointed to see that the projections were by 2030 to be an industry of $60 million only. So just before my talk, I did ChatGPT check, and it was saying the projections now by 2030 are $500 million. So dramatic increase over the 60 million that I found a couple of years ago on the internet. So beyond the point of what exactly is the number, and I don't think anybody can predict accurately, the phage therapy and the whole overall phage industry is picking up in big ways. These are some future directions. That's my last slide. We're looking in more detail into microbial interactions and delineating cytophin communities with, through the observation that the vast majority of mice do have metabolic deficiencies. We're trying to explore this further. We're trying to delineate the mobulome, not only phages in the viruses, because through the identification of actually several million circular genomes from metagenomic data, we do identify potentially completely novel categories of mobile genetic elements. That will be plasmids or viruses, but there may be something completely new as well. We're expanding the networks with the CRISPR arrays, not only with viruses that they presented, but by connecting also novel MGs and plasmids. through CRISPR as well. And finally, an area I haven't discussed at all, which is discovery of novel metabolism and completely novel metabolic pathways, again from metagenomic data. And I'd like to end with looking forward, let's say, slide, which is where we're going to be in 10 years. Chris is one of the best persons to actually answer this question, but here I have another gentleman who is trying to do that. Anybody recognizes who this guy is? His name is Martin Cooper, and he did something incredible, something that changed actually the world back in 1973. He did the first cell phone call. And how is this relevant with what we have been discussing? So, in a recent interview, which I think was incredible, he was asked if he could have predicted the evolution of the cell phone through the times. And his answer was, who could have predicted to put a Kodak film in a cell phone? And why did he say that? Several of you may not know what Kodak film is, but before the digital photography, this is what we were using. So back in 1973, there was no digital photography, right? There was no internet, there was no email. None of the things we do today in a cell phone existed back then. So, and this actually makes you think that we're, not only when we're trying to predict the future, we don't really take into account all these additional areas that are developing in parallel, but this is extremely important for our field as well, to always keep an eye that we do need to connect with all of the other technologies, not just other disciplines, let's say, but other technologies that are constantly coming out there and could completely change the field. For example, before coming to the conference here, I never thought we could apply sterility test in a soil sample. Why? You're trying to find the most dominant. But that's what Venkat suggested. Actually, you could come down to the rare biosphere by applying something that seems completely counterintuitive, which is trying to sterilize the sample and then look for whatever is remains there. Anyway, with that, I'd like to end and thank all the people that have contributed to the studies I have presented. Thank you.
So we'll be opening up for questions, but for time's sake, we'll have one question per person policy for today's session, and you can have more questions outside during the coffee break.
Can it be not better again?
We're starting with Maria.
Okay, here to the right. So I'm a little like skeptical about the virus host. Like I'm also in a virology lab, and when I talk to them about virus and mags and all that, they don't believe anything. So I'm thinking, how are you, or if you guys are thinking, when you say that a virus has a host, you are omitting so many steps into how a virus infects a host. So we're, in order for the virus to actually like get into the genome, if it is going to get into the genome, it needs to be able to infect a specific host, a specific cell. We're thinking that it's not like we don't know the receptor that it uses, and it uses it like. So basically here, it feels like we are omitting that part of like the receptor getting into, and more like, okay, this virus is like found in the genome and that's it, and we're not really counting into like the previous steps of the virus infection.
Yeah, very good point. But in order to get to the question you want to ask, you need this. You need to be able to know that this virus is infecting this host. And then once we have this database of all the virus viral host connections, we can actually start, and this is an area we're trying to get into, but it's extremely difficult, which is trying to do co-evolution of the host and the virus in order to identify potential proteins that play this role. Identify candidate genes from the phage and the host that could be actually responsible for this interaction. So this is very fascinating and incredibly difficult field. And you are asking the hard question here, yes.
Thanks.
Great talk. Thank you, Nikos. Understood, buddy. A common question is sterility issue, the sterility of the soil, according to that. That's an old Japanese idea for isolation of bacillus and rare bacillus species with temporal sterilization, just as an aside to the Japanese public. But my key question here has to do with whether or not you've looked at anti-CRISPR systems in response to this, because that would further corroborate your discussion about the diversity of CRISPR interactions, because bug like F. prosnitzi with 1,100 pins, that's a lot of pins. So the anti-CRISPR, would you say that could give us more indication about that?
Yeah, we have been, at least in the human gut cell, we have been trying to find anti-CRISPR, and in the viral infection networks, we have found a lot of viruses connecting to CRISPR systems from other viruses too. And there is some sort of protection of the host. That's what we see, the virus that infect a specific host. That host is never infected from the virus, that the phage from the virus that attack it from the space around the virus that is connected to that host. So we are calling it infection, but I also agree it may not be infection. Yes, it may be several times phages work like plasmids, right? So they live with the host and they can be integrated and they add additional capabilities to the host. There are genes that can allow us to distinguish between virulence, let's say virulent phages, and that's extremely important as well if you want to use them for phage therapy. You have to identify which are virulent, what are the genes and the overall capabilities that will enable a virus to kill actually the host, right, versus living with the host. So all these are completely new and these are extremely exciting areas to actually work right now.
Okay, let me wait for Chris to ask.
Real quick question there. Just since you can predict the host, basically, could you do a regression of how many possible hosts there are? Answer the question of how many species are in the world potentially.
So how many hosts do not get infected by, yes, we looked into that as well. We couldn't come up with any correlation. So try to see, we found hosts that are never infected by any viruses or by hosts that are infected by some but not by others. This is enormous collection of data. We haven't touched into that, so but the data will be there available. And
Curious.
yeah.
So, you're done?
Yeah.
Okay, so I'm not going to ask you more questions that you said. I will talk to you later, but the one burning question is the following. We had, based on our metagenome, we had more bacteria and archaea compared to virus in clean room. How, based on your experience, because you might have the source from where this kind of viral max that you have got, what is the scenario in your hospital? Is it the same like clean room, or do you have that kind of?
We haven't zoomed into that. But I'm not surprised from what you say. Of course, you can come up with any explanation for anything you can observe. That's the fun part. But it would make sense to me not finding phages in the clean rooms because the phages, most of the phages will be attacking the most abundant main organ. It's not the rare biosphere, right? So if you eliminate most abundant organisms, you will probably get rid of the phages too. That's a wild prediction, but what hypothesis.
Hospital, you should look into that.
Yeah, again, depends what remains after all this sterility and the X-rays or the UV.
Yeah, hi. So I thought it was pretty interesting that when you look into the max in the beginning, you only find like 5% of them correspond to isolates, right? And you're making like quite a important hypothesis out of that. So I was wondering, did you do any double checking for, say, maybe your max were chimeric, right? Like I assume your ANI threshold is pretty high to identity. So did you like check if they're chimeric or maybe like one MAC maps to two different isolates or anything like that?
So, that's a very good question. I did mention, we cannot really use ANI unless we use the high quality marks. So if we drop down to 50%, 60% completion, ANI doesn't really work well. So we use only high quality marks for this conversion, but there is a second line of support into that, of evidence, second line of evidence, which is we have seen that the vast majority of marks do have metabolic deficiencies. So that would suggest again that they are not culturable. So they cannot be isolated. Now, what I didn't discuss, and I think this is probably the most interesting part, is that if this is the case, this dramatically changes the whole industry, because the whole industry is trying to create probiotic industry, let's say, or any industry is trying to cure disease through cocktail of microbes. They're doing it with a cocktail of isolates. These isolates never will live together, and we know that. They die in a week or 2 weeks. They don't want to live together. And what this suggests is that if you want to have a stable community, the only way to do it is through a syntrophic community. So have metabolic deficiencies and partnership. We are not there yet, but I think we're going to the direction of the engineering communities. Imagine that you can take a cocktail of isolates, destroy some metabolic pathways, and force them to become complementary. I think that could be happening in a few years, engineering communities of isolates. But by forcing them to become syntrophic rather than living independent. That's I think the direction we're taking. If the hypothesis is correct.
Simple question, since CRISPR arrays often target plasmids.
Yeah.
Have you done the bridging to find broad host range plasmids or the host plasmid network?
Yes, yes, we've done that too. There are some broad host range plasmids, very few, but there are, and that's the interesting thing. The same way that there are some broad host range viruses. We are seeing very small number of OTUs that they are infecting different phyla. We are very cautious about that. We know that the spacer virus connection is very specific, 35, 40 base pairs, let's say, identity with one SNP is not random. We are certain of that. We don't see hits everywhere. But infecting different phyla, that's a little bit.
There's a known plasmid that affects eukaryotes and bacteria, so I'm not surprised.
Yeah.
Hi, yeah, very nice one. So I saw you have more mags in IMG than NCBI. So are those available for public use?
Actually, we have less mags in IMG than we have they are in NCBI, and you can download it. Yes, they are all packaged for a single download. Sorry?
Your Venn diagram is showing is really high in that.
No, the metagenome part, the max is bigger, the size is bigger in NCBI. However, unfortunately, NCBI max, the vast majority don't have a quality flag. We don't know if they're high or medium quality, so we took everything. And then maybe medium, a lot of them are medium quality as well. But IMG max, there is only high quality.
So it is reprocessed. So the data is saying, that you reprocessed, that's why IMG had more mags, your Venn diagrams were bigger.
No, IMG doesn't have more mags than the SPI, less mags. The gray part is all the use from the genomes. No, we haven't captured from with mags. That's the rare, what I mentioned, the rare biosphere. Yeah.
I do have one question. Oh, yeah, sorry. Yeah, so you did mention that you're thinking of validation approaches for multiple of the things that you did. I have also seen like a few other papers that, especially the ones which released all the MACs and the BDCs and all. So what are the validation pipelines you're thinking of? Are they going to be computational or are they like more experimental based validation?
Yes, validation for the quality of the mags, for what?
Yeah, so like for looking at the metagenomes or the MACs, I mean, all of them have like a lot of false discovery rate also, the current protocol pipeline.
Misassemblies, you mean, or?
Yeah. It can be misassembly as well as quality issues because contaminations and.
Yes,
Yeah.
so yes, for the contamination, we're using, again, we're using SecM, that's SecM tool. This is not the ultimate truth, but it's the best tool we have right now, and that's what the community is using. So with again, with a grain of salt, understanding and realizing that there may be some errors there. For misassemblies, I agree also there could be misassemblies as well. That's why we don't particularly trust marks that single mark OTUs, OTUs that have one or two maybe marks only. So if you want to have more, if you want to have higher confidence, if you go to marks coming from several different samples as well that create the same OTU, then we have much better confidence that these are real. Same with the protein families. If you see a single gene, it may be bogus, but if you see large protein families, the gene is real.
Thank you. Yes. Okay, oh, there's one more question here.
Okay, thank you for your beautiful work. I have only one simple question. In the viral infection network, the nodes are the virus. I want to know what's the editor indicated?
I'm sorry, what?
What's the editor indicated in the viral infection network? The editor between the editor link the virus.
Oh, that's spacers with viruses. So yeah.
Yeah, I mean, what's the mean of the edges between the virus? Yeah.
Yeah. So if the nodes are viruses, then the edges are the co-occurrence of those two viruses
Co-occurrence.
in CRISPR arrays,
Yes.
either on a contig or in the same CRISPR.
Oh, yes, thank you.
I'm sorry.
Yeah, it's okay. I heard you both almost communicate.
So let us give another round of applause. We will now disperse for a coffee break and we'll reassemble at 3:45.
Yeah, I know. I was going to ask that about viruses also, because that was really messed up.
Sorry to pull you. Run the home division. Do I have one?
Great. If you see email, I'm gonna go on. We'll catch up. Why don't we make one?
That's amazing. That's what I think. That's what I think.
That's all right. But is that how do I go backwards? Easy. Thank you. I really appreciate it.
Thank you.
Of course, and,
Yeah, no problem.
we sort of have a smaller group, so we can stand Yes, I'm, yes, the director. But I just, but I'm specific, so my last year. Yes, oh, you found out.
Are you going to a
Yes, okay. Oh, yes. Yes.
So I know all about where are you from?
We've been around the world.
Well, I Singapore from Melbourne. China, Singapore, I'm always knowing something busy. I'm just traveling. I'm off to and then I've got a couple of days and I'm going to New Zealand.
Well, you are diving.
I'm from New York, so we have like.
Yeah.
That's what you're. Yeah. Oh, yeah, we planned this. Everyone.
It's amazing. Thank you very much. Thank you very much. Yeah. Excellent. Thank you. Well, it's a little bit of swag. It's not right. There's one. I'd like to start a presentation with a question.
Yeah. First question is the
First person to put their hand up and answer. I'm sure everyone.
No, I'm gonna get back to you about the Cosmos samples.
Yes.
Yeah. So we'll make sure everything is either in docs or
Yeah, please don't.
Okay, yeah.
Well, you see. Yeah. See, you over yes, right? Oh, actually, don't go straight.
I'm sure I'll be in another meeting, if they want me to.
Yeah.
Yeah, every month I'll call, yeah. Hey.
I'm all ready.
Figure out.
The others give people for minutes.
We have a new one.
Hello.
How long?
That's right. It's like a bit of and now I'm doing
So how long have you been here? PhD in Australia is 3 years. If you don't get it done by 3 years, get out.
Oh, that's
So
not
it's like
US.
6 and a bit years.
No, I was in master's and now I'm just starting.
3 years. And it's like literally by 3 years, it's like,
But that's Europe.
yeah, kind of.
Yeah.
It's like kids at home, right? By a certain time, adults are like,
I think you have qualified.
no, I'm talking about PhD. Like, they'll just keep doing more and more stuff.
So in US, the first 2 years are like research. So it doesn't matter how
Right now, I'm
just having classes, I'm waiting to like sometimes mostly in like public colleges, but not in private ones, and Cornell and other Ivy Leagues already think themselves to scholars and they even let me convert it from me having masters from them.
So you've done the 2 years of masters, and now it's going to be 3-4 years, 5 years.
I started my course last 3 years, so let's see.
What amazing project is that? It's Miriam. It's Miriam, right?
Yeah, this is the chats. See that
She's working on endocrine microbiome, relationship to endocrine development. Maybe. Hang on. I'm shocking. Good with them. No, I'm shocking with names. I'm good with them. We were just testing.
So I work with the microbiome.
Well, because it
Okay, I'll try to bring everyone back.
Try to keep these. Well, that's the thing. Just.
Oh, I'll give it a buzzer like 3 minutes.
Good. Yeah, it's 15 minutes, but I'll try to get through it.
Yes. So I'll give you okay, I'll bring people back. Yeah.
This is live stream, yes. Yeah. And it saves automatically to our channel, so you can like go back.
Hi everyone.
Just remind me about mine.
We're not going, right?
Hello. We are coming to start the second half of the afternoon session. So, can you all please settle down so that we can keep it tight and enjoy Tokyo tonight? Okay. Yeah. Oh. Everything okay? Yeah. Okay, so now we have Mr. Mark Dupal, who is the Vice President of Sales of Twist Bioscience. He has excellent track record in developing and growing business and pharma companies. Today we'll hear more about Twist Bioscience directly from him. And if you have not checked out the counter, feel free to meet Mark outside in the Twist. Thank you.
Thanks very much for inviting me. I'm really privileged to be speaking to a group of people who are far more intelligent than I am. So, is it?
Sorry.
How's that? There we go. Okay. So, thanks for allowing me to be here. I'm lucky enough to run Twist Asia Pacific, which is everything from China down to New Zealand and all the other countries in between. So, we've been given a chance to present. I'm not going to make this salesy, and if I do make it too salesy, you can boo me or you can cuss at me, whatever you want to do. So I'll try not to make it too salesy. But I do like to start my presentations with something a little bit different because I've got this amazing pair of socks. There's only one pair in Japan at the moment, and some lucky person in this room is able to win it. But I do need the first person who can answer this question correctly will get them. So hands up who knows what similarity between Subways and milk, which is a bit random, but it'll make sense later on. Anyone? Anyone? Do you know
Their smell changes over time.
Oh, that's too good an answer, but not quite that fancy. Anyone? Well,
I don't know.
that's kind of good enough. But amazingly, both contain bacteria and viruses. No? And I will make it for you. Enjoy. Enjoy. And although quite a weird one, it'll make sense later on. But I guess as part of what's going on here at MetaSUB is that there's got to be some kind of detection path down the endo, you would think, right? You don't just go and sample all this stuff for the hell of just sampling this stuff. So I'm sure it's for the better of human development. But who's Twist? So we're a company that makes DNA. That's how we were founded. Founded in 2013 with some good ideas, IPO in 2018, and now we've got a couple of manufacturing facilities, so we can make 16 million oligos a day. So we've got huge throughput. If you need DNA, we're pretty good at it. So this is kind of how we differentiate ourselves. Most companies will kind of make DNA in a well or on a glass slide. The way that we kind of change things a little bit is we use a silicon platform, no bigger than an iPhone, and on that we fabricate up to 1 million oligos. So just think about a really, really expensive printer, inkjet printer. That's kind of what this is. But an inkjet printer which costs you a few $1 million. But on this printer, you know, we have these kind of chemical wells. Each chemical well contains 121 unique oligos that we can make up to 500 base pairs long. Back when we first started in 2013, people thought that 200 base pairs was as far as you could go. So we make these really long, really accurately, and in very, very small amounts in the femtomolar scale. So that technology fuels our businesses, and we've got 4 different businesses: drug discovery, data storage, synthetic biology, and next-gen sequencing. And really for us in the NGS space, it's about target enrichment. So what is target enrichment and why target enrichment? So as the name suggests, target enrichment is you have a specific target of DNA, Twist can make you a panel, and then you can go from a pool of DNA that you collect in a subway somewhere, you can go in and you can go and target that DNA. And again, we provide full workflow and sequencer agnostic, but then why would you use target enrichment? So the good thing about target enrichment is when you're designing a 120 base pair piece of DNA to target your specific area, it can tolerate mismatches. So as your bugs start to evolve, if there's not a huge change, up to 10 base pairs change can be tolerated. So you can start pulling down specific bugs that are evolving, and we've got some examples of that. It's pretty flexible, you can upgrade, so you can always evolve your panel. It's never fixed. Scalable, you can have 100 probes or 100 million probes if you want. Right? It really just depends on what you want to target. It's quick, so you can do this within 24 hours from end to end. And the good thing about target enrichment is because you're not sequencing everything, you're only sequencing what you're looking for, the cost is significantly lower. So when you get into a clinical assay, cost is super important. Right? And to even reducing the sequencing cost in these days, in China, you can get a gig for $1. Right? It's even important at that scale to keep it less than $1. So that's why target enrichment also kind of helps. I swear this is my last sales slide. What do we provide? So we've got NGS panels, we've got full library prep, custom panels is kind of like our thing, it's our jam. We've got off-the-shelf panels, so I'm going to be talking about comprehensive viral. This targets about 3,000 known human viruses, so pretty much all known human or mammalian viruses. There's a respiratory panel with 29 different respiratory viruses, AMR. We can make synthetic controls for you, so custom, and we've also, we release them. So we've got influenza, and we've just released the H5N1. Okay, so you guys are collecting tons and tons of data, tons of it. There must be. But what do you do once you've got it all? So I'm going to go through a couple of examples where a lot of data has been collected previously, and how do they use target enrichment. And one of the areas is surveillance, obviously, surely very similar to what you're thinking about here in the future. but there's been 2 papers released quite recently using that comprehensive viral panel. So one of them was looking at looking at rodents in the Yunnan province in China, and trying to understand the virome. Because what's interesting is that if there is another massive outbreak, God forbid, touch wood, you'd like to know what's in the natural population. And so this team took 128 rodents, they looked at different organs and they wanted to see what were the viruses within those organs. But really what they were looking at is, depending on the pollution created by humans, what was the diversity of that virome within those rodents? And what was conclusive is that the more pollution created by the humans, you had a much larger, much diverse amount of viruses in there. Ones that they kind of worry about, because we are really pushing those viruses to probably become more pathogenic over time. Let's hope not. Another one came from Texas, where they're looking at water surveillance. So again, you've got a panel which is targeting over 3,000 viruses, but they're just looking at a couple of different viruses. And they really use it more so during the influenza season, where I think they look at about 17 different influenza serotypes over time. But what they found in 2024 in March, they started to see this peak of H5N1. And they're like, uh-oh, H5N1 is now in our wastewater system. How is it getting there? And so what they concluded, and I don't think it was absolutely conclusive, but what they thought it was coming from was unpasteurized milk. So they identified that it either had a bovine or an avian background, but they think it was coming from unpasteurized milk. So these guys are like, if we need to survey over time, we should be doing it because we've got to know what's going on, and we can do it through wastewater. Okay, so environmental DNA. So why the hell did I ask the similarity between milk? Okay. Farm management is going to change because we can monitor DNA. What's interesting is in cattle, there are a few pathogens that can completely wipe out herds. And you're talking hundreds to thousands of cows, which can cost millions and millions of dollars. So the earlier that you can detect this, the better that you can manage a farm. So farm management will completely change over time. We work with a very large group out of New Zealand who have millions of cows under their remit. And what they look at over time, and what they've done is they've taken 16S and just done metagenome sequencing of the milk itself. Milk is a great indicator of what's going on in a cow. So we built them a panel. We were looking at 400 very specific species. There's about 67,000 probes. We tested on 5,000 cows. And what you can see here is this is the amount of sequencing or reads necessary in target enrichment versus the reads that they would use for shotgun. Concordance was good, but they used 1/10 the amount of sequencing. So when you're on a farm and you are looking to sequence every cow over time, that reduction means something. And so here's a real life example where cows are stoic, right? That cows don't really have much emotion, and for a good reason. Because when they're in the Sahara or wherever they are, they don't want to be eaten. So when they're sick, they don't want to show they're sick either. And so it's incredibly important to look at the health of cows through their life cycle. And what you'll see here, what was really interesting, this is the month, you can see the year, and all of these are a different bacteria. But one of them, Nocardia, which causes mastitis. So once you have mastitis, it effectively stops the cow from being able to produce milk. A farmer rang the person from LAC, they said, we've got a problem and we can't get rid of it. We're gonna have to get rid of the cows. Right? So we're talking about 150, 200 cows on this one farm. They went back through the data that had been collected, and what you'll see, so this is Nicardia, and what they noticed is, okay, in September, basically from June to September was no good, no trace, October, nothing. Then all of a sudden in December, they started seeing it. January, it got higher. April, it got higher. Although they weren't actually using this data because it was a trial, when they went back, they could see that early detection, they could, if they were testing every month, they could have picked it up early. They could have treated it with just some cheap antibiotic and saved this herd and hundreds and hundreds of thousands of dollars. So farm management is going to change because of things that you're doing and picking up in the environment that they can then really use in a sense which allows them to manage cattle, feed, everything. It will completely change in the future. This is my final slide. So we've also got a customer who is now collecting environmental DNA, but through air. So you can get these air canisters as people are coming off planes, you can collect the DNA, and then you can sequence what the hell is someone coming from God knows where, what are they bringing with them? You can detect what the air is like at different times of the year, etc. So, I'm sorry I don't have the sequencing data yet, it's brand spanking new, but this is one of our new library prep kits, and from as low as, and now that it says here 0.5 nanograms, we've now got data from them that shows that 0.3 nanogram total DNA from a canister, you can produce a library. And this will be able to go onto a sequencer and sequence what the hell is in the air. So, and this is the thing, it says cell-free DNA, it is focused on cell-free DNA, but it varies, it's a high efficiency library prep that will, with a specific ligase that we've engineered, that will allow you to really take everything and just ligate and be able to produce something. So, that's me. Was that on time?
Yes. Thank you. We are now open for questions.
It's not nice, Mark. Two questions. One is, how is this different from AmpliSeq? You remember AmpliSeq?
Yeah. So AmpliSeq is an amplicon-based type technology. So you would use target enrichment when you got more targets.
This is hybridization based.
This is hybridization based,
Okay,
exactly.
That's good. So the second question is, how do you validate whatever you are saying is positive? Can we pick up from your panel and then resequence it? Or
Yeah.
you have to go back to eDNA?
So no. What happens in the example of the milk? So they have specific, bacteria that they can serially dilute, and we with them tested. We could detect a single colony forming unit of bacteria, but they've got that. We hybridize, we capture, we amplify enough. It goes on the sequencer, and you can sequence it.
But you cannot take it from your panel and resequence.
The panel just captures whatever is actually in there. So
That's fine.
yeah. Thank you.
All right, thanks for the talk. Real quick, so for surveillance, a lot of wastewater surveillance groups in Europe rely on qPCR-based identification, for instance. I've tested Illumina respiratory pathogen, and it's not the greatest. So we're testing your panel, and it's the same basic technology. The amount of manpower or number of hours required for the hybridization protocols is pretty high. Is yours any better?
So we have two protocols. There's a fast hide, which can be done in about 4 hours. But I would say with wastewater management, if you are looking for something that is obscure, you probably want to do an overnight hide. So you're talking maybe 2 days worth of work to be able to pick up. And there is a difference between the way that Illumina does it and we do it. There's, when you build these panels, DNA reacts differently. So you may have one probe which binds just exquisitely. You may have another probe which is not binding so well, right? So what we do is we can boost the tiling. And over thousands and thousands and thousands of iterations, our algorithms just got better. And that's kind of where we differentiate. It's that boosting which gives you really good uniformity, so you're not over sequencing something. Kind of sequence is the same. Hopefully that answers the question. Yeah.
Thank you.
Thank you.
Up next, we have Dr. Alexander Lukaci, who earned his PhD in bioinformatics from Temple University, where his doctoral research was centered on the development and application of evolutionary model. In 2023, Dr. Lukaci joined Weill Cornell Medicine as a postdoctoral associate. His current research encompasses a variety of projects that include evolutionary modeling, infectious disease, and cancer, with a particular focus on the intersection of these fields.
One moment, everyone.
Hello, everyone. Glad you are all here. Thank you to the organizers. Thank you, Krista. Thank you to everyone else in the lab as well for organizing this fantastic event. My name is Alexander Lukaci. I'm a postdoc with Chris Mason, and I've also recently assumed the bioinformatics responsibilities of the MetaSUB consortium. Today is going to be a pretty gentle introduction into evolutionary modeling. The talk is entitled Quantifying Natural Selection Coding Sequences. I'm presenting general purpose evolutionary models, but also tying this in at the end with the work of the MetaSUB consortium and how these models can be used to profile cities and to understand the urban microbiome. I wanted to begin by discussing additional use cases of these types of evolutionary models. So one of the large scale, high profile work of these types of models has been to create mutational profiles of spike region during the COVID-19 pandemic. These have also been extended to other additional variants of concern and interest during that period of time. But you're able to detect in sort of the upper left corner sites of important mutations within important regions of the protein. You're able then, in panel B, to track these mutations over time to see how selection and drift, but mostly selection should fix these mutations in the population and how these vary over time, especially across both spatial and temporal components. Figure C shows different levels of nucleotide diversity in different types of regions, either in the whole genome or spike specifically. Ultimately, though, once you detect these types of sites using these models, you want to correlate them or annotate them with functional importance. And you can do this by using protein structure, look at specific regions where these mutations occur and infer some type of relationship in the function of protein-protein interaction or other types of important physiochemical properties or other important properties of proteins. They're not limited to just viruses. So I want to again reiterate that these are general purpose evolution models. These can be applied to bacteria, as the consortium has abundance of these types of data. You can look for recombination, so the figure on the left, in bacterial species. You can profile the entire genome of these types of species and look for evidence of selection across different proteins. These are what you see on the right side with spikes for different types of proteins. Again, these are general purpose models. My whole department, my PhD is mostly ecology and evolution. So we have a lot of people interested in birds, lizards, all types of species. So these types of models can be used to profile lineage-specific, so branch-specific evolution of important proteins, in this case, the immune system. These models have also been applied to cancer, so work by David Posada and others have applied these types of evolution models to look for not just drivers of cancer, but also negative selection, things that while they mutate, they preserve their functional properties. Obviously, most genes, most of the time, are under neutral selection or drift. We're looking for evidence beyond the null hypothesis of neutrality to make these sort of claims. Again, it's not just limited here in my own work, in collaboration with a postdoc that was at Cornell at the time, we profiled the evolutionary history of BDNF, which is an important brain development protein, right? This protein is older than the dinosaurs. And we did the deepest comparative genomic analysis to date and tried to relate selection on this protein in regulatory regions. This is actually expressed as a pre-pro protein, it needs to be cleaved into functional form. And we showed that while the core functional domains, the NGF defined domain, are under negative selection, these regulatory components are actually under some evidence of positive selection that are species specific. Ultimately, this relates to a number of neuropsychological disorders, psychiatric disorders, including schizophrenia. We've also profiled another protein, this time mammalian REB2, which is a GTPase. It's responsible for regulating synaptic development, also controls the intrinsic excitability of neurons. We again applied evolutionary models to scan across the protein to look for evidence of selection. At this time, we applied a ancestral sequence reconstruction approach using these models and inferred the protein structure of not just the species that exist today, the sort of extant species, but inferred these extinct species. And we're able to show when the protein evolved along the phylogenetic tree, able to tie that in with changes in protein structure. So it's married both sequence analysis and protein level selection. Additional work in other proteins. I've also worked in heat shock proteins expressed across kingdoms of life. Very deep importance in very, very fundamental foundational properties of cells, right, for chaperone and stress response, these kinds of things. We're able to compare now a family of proteins, look for selection across all four of these members, B1, B3, B5, B8, and able to make statistical comparisons to show which protein in this family is evolving at the fastest rate in comparison to the rest of the group. In this case, you can see an elevated rate of the NDS in the HSPP3 protein. Again, general purpose models, I want to really hammer this home. We've also done work in plant biology. We actually looked at the genome. We had a collaborator in Sao Paulo that goes into the Amazon rainforest, in this region called the Pantanal. They did the genome assembly of a new species of carnivorous plants. Actually, then created a phylogeny of mixotrophic species, like plants that are able to eat insects, carnivorous, and photosynthetic, and compare that to just photosynthetic plants, and able to profile the chloroplast and mitochondrial genome to look again for evidence of selection. We've also applied these models, again, general purpose. We've had a number of talks today about the marine environment, right, from corals to fish. We also have been applying these models to look for how endothermy evolved in fishes. We again did an analysis where we compare fish that are endothermic, which there are not many, but there are some, and able to compare that against the genomes of non-endothermic species to again look for candidate genes that are under selection in fishes that have endothermy versus fish that do not. When I'm describing these evolutionary models, I'm describing decades of work and trying to understand evolutionary processes. Typically, when you have sequences, you'll do something like Hamming distances or proportional distances. These are typically how a lot of these distance-based metrics have begun, right? What I'm really describing, though, is seminal work in 1969 by Juice and Cantor to put these types of sequence-based distances into a Markov process and create a sort of probabilistic framework for these types of things. This model in 1969 was obviously simple, based on known biology at the time. But as we've learned more about biological processes and important properties of biology. We've gone back and refined the models, adding additional parameterizations for things like transversion bias, compositional bias, and things like GC bias, different types of rates for purines and pyrimidines, and reversibility, ultimately getting to a point where these models are now parameterized the entire distance space from sort of, from a nucleotide to a nucleotide. However, when I'm describing quantifying selection, I'm really going to be talking about things not from the lens of the nucleotide, but from the lens of the codon, right? So we borrow this, the field has borrowed a metric from population genetics and has created this metric called DNDS, and has a simple interpretation, right? When the value is less than 1, you have negative selection, you have preservation of the protein function. When you have 1, you have neutrality, and when greater than 1, you expect adaptive evolution, evidence suggesting adaptive evolution. And you can see this at the bottom. You have some evidence of sites that are colored white, that are under constraint, while there are mutations there. They're preserving the amino acid at specific locations, and in pink you see different sites that are colored, that are fast sites that are changing the amino acid residue at a specific location. These models are generally encoded in large matrices. Bioinformatics is full of matrices. And this is no exception. Many of these are parameterized based on the, from and to codon, with the different parameters based on the codon table in terms of are these changing the amino acid or not. Most of this matrix, though, is encoded for only single hits, only single nucleotide exchanges. What that looks like is here on the left. When you have an occurrence of a multiple nucleotide substitution, it assumes that these take multiple series of mutational events. However, again, we've learned more about the biological process. We know there are things like aerochrome polymerases that can introduce multiple nucleotide substitutions in a single event. So we've gone back and we've reparameterized those models to account for this biological phenomenon, and I spent my PhD developing and validating these types of models. In terms of the history of codon models, codon substitution models, this is actually foundational work by Japanese scientists, Masatoshi Nei and Takashi Gojibori, who created a simple method to look for expected values of changes between codons. So they created an expected value based on the genetic code, where for any given codon, in this case GAA, if you have a random mutation, what does that result in? And a third of the time, you have synonymous exchanges, and most of the time you have non-synonymous exchanges. And what you want to do is that establishes now a baseline rate for you, and when you observe changes between two sequences, you want the observed number of differences to be higher than your expected values, right? You're sort of gaining evidence that selection is acting in one way or another over random mutations. However, clearly, these are seminal. This is seminal work, but simple models, right? Simple models sometimes add complexity to account for different properties of the evolutionary process. In this case, this underestimation as sequences grow higher in divergence. And I want to go on a bit of a tangent. I'm sure MetaSUB style have more slides than time. So I want to describe a little bit the life of Nei. Nei is really one of the founding fathers of molecular evolution. The field would not be where it is today without him. He was born in Japan, really a humble beginning, and got his PhD from Kyoto University. Many of his students are very serious evolutionists in their own right. Here's a picture of just some of his early students. He published hundreds of papers, several books, which are really important, important works in the field of molecular evolution. Also started MBE, probably the best journal for evolution, and the society that governs this journal with Walter Fitch, who's again another extremely important figure in the field of evolution. And what I'm really trying to get at is science is fundamentally a human business, right? Humans are doing this work. We want to connect with who these people really are. Photos from the lab, so from the 90s, so 20 or 30 years from now, we'll be able to look back at the MetaSUB photos as well. If you've ever used Mega software is extremely popular for molecular evolution and phylogeny. This is Sudhir Kumar, who's the main developer. Ultimately, Masatoshi Nei won the Kyoto Prize for really a lifetime achievement and really lifetime of work in evolution over decades. I now want to discuss sort of that's the early history of this field, and I want to discuss sort of the modern view. Modern view was created in 1994. It's a bit of a developed, mature field in that sense, comparatively. Spencer Muse, Brandon Yao, and a separate paper by Nick Goldman and Xinyang Yang in the same journal. Back to back. You can kind of think of these different models as sort of the Pepsi and Coke variety, right? Same ideas, but parameterized slightly differently in their outcomes. These are also markup models. They allow you to encode the evolution process in a probabilistic framework. You're able to compute likelihoods and extend these to Bayesian frameworks. You can account for different confounding processes like multiple nucleotide substitutions or any other types of biological phenomena that may be important, such as physiochemical properties or anything like that. And you can have additional refinements that are still going on today with active development of additional models. I'm going to discuss codon substitution models using examples from 3 data sets. West Nile data set, an HIV transmission pair where we know both the source and the recipient of the viral infection, and a bit about SARS-2 spike protein from the alpha and delta clades. These are what the trees look like based on the phylogenies. You can sort of think about where evolutionary signal is coming from within these types of groups, and how our evolutionary analyses may be impacted by the topology of these sort of associations. Here's a version for a spike. It's also a way to think about what is the information content of your protein. When we apply the simplest types of codon substitution models, we can see that for all 3 cases we have negative selection. So most of these are models that generally average selection across the entire protein. And when you do that, most of the protein is conserved most of the time, so you get evidence of negative selection, or neutrality in this case, but generally you get, it's lowly powered to detect selection because it's averaging across the entire protein. Clearly different sites, different amino acid residues within proteins are evolving at different rates, and you need to be able to account for these kinds of things. So these are what I'm referring to in the previous slide, again, are mean gene-wide estimates, right? You're averaging across the entire protein. And they are useful when you have small data sets, so sometimes we're in a situation, usually we're in a situation where we have too much data. Sometimes you're in a situation where these are rare samples, and it's good to use these types of averaging approaches in order to gain power to infer selection. Here's an example from patients. These are patients with HIV, HIV-positive patients, and you can look at evidence of intrahost evolution. You can look for different proteins, such as envelope, for both a slow and fast rate of escape. You can infer both the rate of non-synonymous and synonymous exchange. And again, what you want is evidence above neutrality. The synonymous rate can be thought of as a neutral level of evolution. And you want evidence, in this case, in rapid escape, of an excess of non-synonymous exchanges in the protein. Whereas for slow escape, you can see that the neutral rate is higher than the non-synonymous rate. When you look at these types of things, if you look closer, if you look for, on the x-axis here you have the residue within the protein, and on the y-axis you have different temporal time, right, like the temporal component to this. So you can think of this, how we can contextualize this in the framework of the MetaSUB consortium. We have proteins of interest, we have cities that are sample form, and they're over time, right? So we can try to apply temporal analyses to look for individual sites that are under selection at this point in time that may change over time as we go up, right? As times change, different residues may be under selection at different moments. Ultimately, we can combine these types of temporal analyses to look for hotspots of evolution. But again, we can also have an option to split that out and look for specific sites that are under selection at specific times within cities. This allows you to create sort of more molecular portraits of cities. I want to describe selection as an image processing problem. So you can think of a multiple sequence alignment where you have sites on the x-axis and the branches you have phylogeny on the y-axis. And you want to infer positions within this alignment that are giving evolutionary signals that are under selection, right? Color is not important here. What you're looking for is mean values of evolutionary rates. Obviously, evolution is noisy and unobserved, even when we sample well and deep and sequentially. We're still missing a lot of the evolutionary process. It's unobserved and noisy. Here's another replicate of the same idea. So it's hard to pick out, there's lots of local variation, but in general, you can still pick out the monkey. You can still start to see global features of this process. So there's high local variability, but there's stable elements at the global level. The desired resolution to get branch and site specific resolution is not possible, but we could pick out some of these global patterns that are there. This is what you're doing when you apply G and Y measures. So what you're asking is, if I just average over every single pixel, which is this bottom corner, is the color of that average sufficiently bright enough to first selection? That's why in those 3 data set examples before, we weren't able to find evidence of positive selection in those because it's lowly powered if you're just averaging over everything. The benefits of these types of methods, though, they're very simple, they're compute light, you can run these very quickly. Again, they're important when there's data sets or sample like smaller size, things like that, or important samples that are hard to get. But clearly there's room for improvement. So there's 2 main messages I want to get through in this talk. That these are general purpose evolutionary models, and that the entire field, the idea is that as we learn more about evolution, we've gone back and we refine these models and make them more powerful. So here's a version of the model now that averages and looks for evidence pooling information across sites and branches, right? So again, we have, think of the pixels again as part of an image, but now you're able to get specific information about specific sites within your alignment, and you're able to pick out hotspots. So for example, if I look at this heat map everywhere, I can find a specific codon of interest that I don't get when I average across everything, right? And I can zoom in on a specific codon, I can see what's going on there. I can look at specific mutational changes that are occurring on ancestral branches or terminal branches to really get a detailed, high-resolution image of what's going on. When you apply these sort of updated, more complex methods, you get an idea of, you get a difference from the sort of baseline averaging models, right? You now have evidence in HIV of strong evidence of positive selection on specific regions within your alignment. And as well as for spike, it changes your interpretation once you're able to create more complex models that are parameterizing the evolutionary process a little bit further. These can also be further extended, right? So this is the base, that is now a baseline model. There's been additional work by some postdocs in the group, myself creating a different type of model that accommodates multiple nucleotide substitutions and extending, again, this work to accommodate that as well. We can also look for branches that are under selection. This is another type of method that now looks for lineage-specific evolution. If you remember that slide that I showed of the waterfowl looking at immune genes, you can look for lineage-specific locations. These could be things like specific cities if applied now to the MetaSUB consortium data sets. You can look for lineage-specific evolution, for different types of locations in the phylogenetic tree based on spatial data that we're able to collect and process. When you have these types of models, you have a baseline model that shows, this is now applied to the HIV-1 data set where we know both the source and recipients. And there's traditional models have when applied here, show a high level of selection on the transmission branch, and I think these two. However, when you have more complex models, you're able to show that selection is not on the transmission branch, but actually may be part due to intrahost evolution of that viral infection. Here's a quick summary of what's going on for this different type of method. It's called ABSREL. You're able to detect specific branches that are under selection over a proportion of the tree, and that varies based on the data set analyzed. Why is that important? Okay, well, here's an example of using a traditional model, the GTR plus gamma model, to measure the date of when measles emerged, right? And using a baseline model, you're able to get an estimate of about 600 years ago. However, we have evidence in the literature, ancient literature now, that measles is at least 1,500, 5,000 years ago old, right, by a Persian physician, and describing the diagnosis of this disease in patients. When you're able to apply more complex models, that changes these internal branch length estimates to make them longer, and that those longer estimates provide more accurate, more reliable estimates of viral emergence. We can also apply these models for interpreting intrahost and interspecies pathogens. This could be applicable for things like the MetaCAX project, where we can now look for interpreting selection on internal branches. Internal branches imply that there's a transmission event. We can also look for selection on the terminal branches, which is more of the intrahost evolutionary component, and then compare and contrast the results to look for differences in the group, to look for differences in in selection based on the immune pressure from the host versus selective pressure from transmission events. And this is what that'll look like when you partition a tree to form that type of relationship. You'll have internal nodes labeled in the tree. You can measure selection using that. You can then go back and label the tips and reinforce selection based on just on tips. There's complexity and there's nuance in terms of interpreting that. Many of these are often dead ends or just intra-host variability that maybe have not resulted, selection has not acted completely. So there's some ways to think about how you parse those types of results. But it's possible using these types of methods, so at least begin to ask and answer these types of questions. This slide I borrowed from Dr. Mesa. I think it's a little old. You guys have seen it, other presenters have also shown this type of figure. We have data from the MetaSUB consortium across cities, and across time now. And the idea is, how can we extend the type of work that the consortium has been doing, providing high quality sampling, high quality sequencing, QC assembly, taxonomic classification. We can take these cities, we can take these data sets and these sequences and now start to think about how we can apply these types of models to create molecular portraits of selection across cities and across time. The idea is you can look at specific pathogens, specific viruses or bacteria that are there, and understand if in some city at some point in time, those pathogens are evolving faster or slower in relation to one another. We've begun to apply these types of models to these types of data using an RERP tree from recent sampling with the MetaSUB consortium to find sites, not only sites that are on a selection in different clades, so this is a specific site. We can look at the exchangeability. This is directional selection, so you're adding a time component. You can look for selection profiles at individual sites for a specific clade. You can compare this to other clades. These are what the other core diagrams are doing. And you can now create a spatial, a molecular profile of these types of sites within these types of clades, and now look at how that's varying across different cities or different regions, right? So the idea is we now identifying those locations are important, identifying the mutational exchanges that are going on in these sites are important. We can then create spatial signatures based on this to show which sites are under selection. So here, the x-axis, and then using geographic information at the country level, we can start to look for sites that are under selection in some cities or some countries that are not under selection in other sites or cities, either for forecasting prediction measures or for other chasing other sort of biological hypotheses. It's the end of my slide. I want to say thank you to everybody. Obviously, again, thank you, Krista, and everybody else. This is an incomplete portrait of the Mason Lab. Quite a few more people. But thank you for listening, and happy to take any questions.
Thank you.
Thanks. That was an awesome talk. A metaphor of the image taught me more about evolutionary biology than I learned in all of my undergrad courses, so spectacular. Okay, I got a ton of questions, but first one is, there's a lot of nuance in there. And as is the case with evolutionary biologists, which is wonderful. Metagenomics people, we are not known for nuance. In fact, the opposite. And that's really highlighted by in gene catalogs when we are clustering ORFs based on sequence identity. Maybe a blossom matrix ends up in there at some point, but it's like usually we said 90% is the cutoff, and those things are the same. How do you feel when you see that? And are there better ways that you would think, okay, just given what you were saying earlier on using gene best ratios, where you compare two proteins or two nucleic acid sequences, like, how should we be doing that? Because it's clear we're not doing it correctly to optimize for functionally united clusters.
Yeah, it's a good question. It's always an issue of QC and alignment, confidence, and it's not an easy task. I think, working at the intersection of fields and bringing workhorse methods from one field to genomics is not always going to be easier or perhaps straightforward, but there are ways to build data sets maybe based on reliable proteins like RERP, right? Start there, have proteins where we have confidence in what's going on, and then extend it out to putative and other ORFs and things like that that are maybe lower confidence, but we can start to build a sort of foundation in that sense. But that sufficient to answer your question, or?
Yeah, I got a question. This is super fascinating talk. I love the evolutionary aspect to it. And just looking at these evolutionary trees, I was wondering if you've ever considered doing a coalescent-based model to see where these citywide microbiomes started to, differ from each other and kind of where they coalesce.
Yeah, no, that's a fantastic idea. I haven't done it yet, but certainly could go in that way. Another approach, once we get to answer to follow on from Brady's point, high quality alignments and higher confidence in our data. You can do essential sequence reconstruction as well to also track these processes across extent sequences and things like that. Look back in the history of specific species or specific proteins within species.
Alex, this is me. It's really a very good way of seeing changes that we are seeing in space station. A lot of time people are asking where it came from or something. Okay, so I think it is Brandon also asked a question. So if a species is defined, basically 95% of A and I between the nearest neighbor. So that means you have 5% of four or 5 megabase genome. So you can calculate this evolution based on that. 5% difference is huge there.
You would do it protein by protein, and the proteins will evolve at different rates. So while the global rate may be 5%, there may be higher conserved regions. Then it's a question of finding in the important functional ones. The alignments may be more difficult to produce, especially the more reliable alignments. But you can try, especially like you may not be, you may not need to align the entire protein. You can do alignments based on important functional domains that may be more conserved than that 5%.
Okay, I'll send you email, we'll talk more.
Fantastic.
Thank you for your talk. Thank you for the talk. Next we have Justin Su. Next we have Justin Su, who is a PhD student at University of Southern California. His work encompasses in microbiology and virology, especially designing methods for studying viruses and their role. His recent endeavor has been utilizing metagenomics to understand the microbiology of wastewater.
Great, last talk of the day. Yeah, thank you for that kind introduction. My name is Justin Su, and good afternoon. Konnichiwa. Justin Su desu. Yoroshiku onegaishimasu. I'm still practicing my Japanese. But I'm a PhD student at the University of Southern California. And I'm in Dr. Sergey Mangol's lab in Los Angeles. And I'm really happy to be here. I'd like to thank the organizers from MetaSUB for allowing me to be here. So I'm representing a team in Dr. Mangol's lab working on this ongoing project, specifically in wastewater. So I actually arrived 2 days before the conference had started, and I was walking, it was very humid, walking in the heat, and I came across this building, and I was wondering if the audience knows where this is, or any of the locals. It is actually, it's actually really close here. It's only a 6-minute drive, but it is actually a sewage museum. It's probably one of the few in the world that actually has a museum dedicated to sewage, and it's located in Arake and over there. And it's actually very nice to have been there just this past weekend. So I'm not sure if these slides are. few slides. Yeah. It's not up from one.
Okay, got it. No worries.
Thank you. So that is me, actually. So thank you. So it was really nice to see the museum and to see actually the town of visible sewage. And for me, it was very hot, so I just strolling around. But I'm from Los Angeles. A city in Los Angeles is known as Beverly Hills, and this is a manhole cover. It's as is for sewage. So I'm not actually strong enough to lift a manhole cover, but we're a lot of people are probably curious what's actually inside a manhole. And I found a photo back in 2007, a relatively clean manhole of insert where we see sewage. It's relatively clean, but and when you open the manhole, there's just a lot of stuff that's going on in there. But as biologists or as I was interested in viruses when I was an undergrad, we're curious to see what's actually in wastewater. And as talks that have been given previously today have shown that we can use wastewater. And during the pandemic, we were interested in seeing SARS-CoV-2, the virus that causes COVID-19, in these wastewater. And we're curious to see how the virus got there in the first place. And it's through the sewage where the virus sheds from the host, us as humans, through the plumbing networks, through the sewage, and it goes directly through the sewage and through the networks to the sewage facility, these wastewater treatment plants. And throughout the pandemic, there were many great efforts in terms of wastewater sampling. And some of these sampling had occurred in these manholes. So we can see that right before it reaches these wastewater treatment plants, before the wastewater gets treated, we can take samples from sewage and process them through sequencing to see what viruses are there, especially for SARS-CoV-2, and how much of it is out there. And as you can see, this requires no clinical interactions. It's just straight from the environment where we can just take a sample. So, but from the sample to sequencing, that there's many considerations to be discussed, and some of them are sample related, where of course genomic background from other species are dominant within these samples. There could be degradation and fragmentation of viral RNA in these samples from wastewater. And with PCR-based methods, there are inhibitors that are present in these samples, and of course with wastewater, different pHs, temperatures, even solids and different metagenomic profiles would contribute to a highly variable sample. And in this case would have resulting in low loads of SARS-CoV-2 viruses in these samples. When we can move into wet lab conditions, through RNA concentrations efforts, there could be usage of different wet lab kits and different protocols that are used across the labs that are collecting these samples. And with downstream sequencing, there are different sequencing technologies out there and different platforms. Some may be short read platforms and long read platforms. And there are different amplicon panels, as we'll dive into later, and also different library preparation kits and different flow cells that are used in these sequencing protocols. So altogether, they create a complex, highly variable sequencing data, and that are often of low quality. And all of that can actually be scaled on a global level. Here is a map of all the wastewater studies that have been conducted as of 2022. And there are also phylogenetic studies also conducted around the world. But we can see that there are these pockets of regions in the world that don't have these wastewater studies as of 2022, and some notably in Africa and even in Southeast Asia. And in Taiwan, where my parents are from, and in Latin America as well. So there are still disparities that in terms of wastewater studies. And when we look at as of today, we have actually still there's some increase in wastewater studies, but these pockets are still there. And this is from an initiative from UC Merced that looks into the current wastewater studies that are being conducted around the world. All to say that there is an urgent need for scalable and cost-effective long-term passive surveillance methods. And as we know from the talk earlier, that active surveillance methods are costly and very laborious, but of course, there is a need for all types of surveillance, especially with SARS-CoV-2, but also other pathogens as well. When we look at wastewater in the genome, we see that there are mixtures of highly similar genomes and lineages of SARS-CoV-2. And when we conduct amplicon sequencing, or specific targeted amplicon sequencing, that we get very complex and variable sequencing data. And but the limitations here, we see that there can be different amplicon abundances in terms of length, and also amplicon dropout, where if there's a mutation when primer binds, you lose that primer binding site. So there could be that. And as we know, SARS-CoV-2 as an RNA virus is highly mutable. And one consideration is also that mutations must be discriminated from sequencing errors as well. So when we get these the reads, we utilize bioinformatic methods in order to call the lineages in samples and also estimate their relative abundances. And there has been, during the pandemic, an influx of a lot of bioinformatic methods out there. And that's where the Mango Lab wants to conduct our benchmarking study and also see what bioinformatic methods are there, where we're interested in. And as I mentioned, during the pandemic, a lot of tools were being made and repurposed, and there's still a growing amount of tools out there. So in our lab, we were interested in benchmarking these tools, And some of these tools, as I mentioned, are repurposed transcriptomic tools, such as Callisto-7, RSAM, and ISONIM2. And then we are considering metagenomic methods such as cracking2. And then there were methods that were specifically designed for subsequent to wastewater surveillance. Most of them are highlighted in blue, are methods that use a reclassification approach, and those highlighted in orange are utilizing the deconvolution approach. And for reclassification approaches, they take the reads that are aligned and essentially assign them to a lineage through maximization likelihood estimation, which are through expectation maximization methods. And these are taking the counts and translating them into relative lineage abundances. And the deconvolution approach utilizes a library of mutation frequencies that are then translated into a relative lineage abundance through the least squares method to quantify these lineages. And each of them, we will talk about. One uses the references, one also utilizes a mutation library database. So this is an ongoing effort, and benchmarking is very important, especially to us in the Mango lab. So we are also opening for anyone who would like to join our benchmarking efforts, because some of these tools are actually relatively difficult to install and benchmark. So this is still an ongoing effort as well, and could be a module for camp as well. Some of these tools. So for our benchmarking studies, we focused on in silico methods where we wanted to generate realistic lineage mixtures, and we utilized two databases, GISID and NextStrain. For GISID, we looked at the counts and that were above 10 and looked at co-occurring lineages from a certain time point to mimic essentially the pandemic era. And we also did, for NextStrain, we looked at the sublineages, which were a mixture of sublineages and parent lineages, and took their relative abundance percentages as well. And we looked at 11 lineages and 12 sublineages and created 42 time points for lineages and 36 time points for the sublineages. We picked one genome per lineage that we wanted to seek out, and essentially inputted those into a universal read simulator called ART. And ART essentially is a next generation sequence read simulator, and it has parameters where it's able to generate simulate reads based on different parameters that are given. And what we have done was that we were interested in generating these data sets of different lengths, different total, different read counts. They may be 1 million or 5 million reads. And we looked at different Illumina platforms of different sequencing rates to compare, and also to include errors, sequencing errors or not, within the simulated data set. So we currently have with the lineages and sublineages a mixture of 78 lineages, 78 mixtures, and our preliminary data, where we took 5 of these tools, and the 3 in the top were reclassification tools, and the 2 in the bottom are deconvolution tools. And we wanted to look at their mean absolute error, where we took the true where we knew the true frequencies and compared them to the ones outputted by the tools. For art, we didn't include any errors, but we used Illumina pair-end reads of 150 base pairs with 100 million reads. And these are our preliminary results, where we see that that we have our read classification tools that, at least for lineages, perform very well compared to the deconvolution tools. And we also have where we see the sublineages where they performed not as well, but that is due to being the data set being sublineages. And this is where we see that RNA-seq-based tools showed higher accuracy that utilizes the maximization, the expectation maximization algorithms, where they were more accurate than the deconvolution methods with the least squares. And all of the methods showed lower performance with the closely related sublineages as well. We also looked at the distribution of absolute errors in terms of predictions. On the left y-axis, we have the percentage of accurate predictions, and we also looked at the x-axis was the absolute error. And for our threshold, when we see that for lineages, 97% of the predictions of outcome had at least fewer than 0.04% error. And for PIGAGs, or pipelines in genomics, that went down to 87%. And we see a decline where, in terms of accurate predictions with sublineages, with especially the deconvolution tools as well. So as this is an ongoing project, we definitely have future directions, and not only do we want to simulate in silico samples, we also want to generate in vitro sequencing data using synthetic RNA genomes. And we wanted to essentially use synthetic SARS-CoV-2 RNA lineage and sublineage genomes as sort of a gold standard for our experiments. And it just so happens that Twist Biosciences is here, that we would use these synthetic RNA genomes and generate the mixtures where we can directly sequence them through different sequencing technology platforms, and where we can also spike in through RNA extracted from wastewater, and also just spike in just raw wastewater and see the results from there. And we have short read platforms and long read platforms with Oxford Nanopore. And we also wanted to use in vitro sequencing data, generate in vitro sequencing data with inactive viral particles, and the same can be done in terms of directly sequencing or spiking in as well with raw wastewater. And one of our, in terms of future works, is looking at a read simulator where that is designed for wastewater properties. And one of them was recently had been on as a preprint as Swampy, which they looked into, which they are capable of looking, having different parameters that would be suited towards wastewater, such as variable amplicon abundances, where they can generate that, and also parameters for errors in wastewater due to RNA degradation, and introducing PCR errors and also library preparation artifacts is something that they consider. And essentially what they have done is wrapped art into it, where we can essentially use, look at quality scores and look at different sequencing device errors, where we can put that in as an input, as a parameter. we have essentially seen that there are certain, it's still an active developing simulator, so we're looking into this, and we're also looking into other in silico generation of reads as well. So with this, I would like to thank my PhD advisor, Dr. Sergey Mangel, who really wanted to be here, but due to certain scheduling where he's not able to. But I'd like to thank the team in our lab with Viriel, Victor, Dr. Sharma, and Dr. Torres as well, and Dr. Martin Holzer from the Robert Koch Institute in Berlin, Germany, and also from the University of Miami, Dr. Helena Solo-Gabriel, and the Smith Lab at USC with his Dr. Smith and his grad student, Michael Saldana. And the entire project team, which I will briefly take this time to talk about where I was this summer, which has actually with Dr. Lucaci. We were in Romania just a month ago, where I was, and we participated in this this school called the Eastern European Bioinformatics and Computational Genomics School. For next year, it will be in Krakow, so with Dr. Lepage. And so we are interested in seeing if there are any sponsors that may contribute to being a part of this school and growing effort school. Yes. So with that, I'll also thank the organizers again, MetaSUB and Krista, who this is an amazing conference and that I'm really proud to be a part of. And of course, Dr. Mason for including me as well. And definitely the sponsors for making all of this possible. So thank you for listening, and I'm happy to take any questions.
Thank you.
Hi, thanks for the talk. You notice from your map, you left out Scandinavia. A lot of Europe actually has formalized sampling with wastewater, with sequencing every week. So we might want to look into what is freely available, but not necessarily published. So I think that's something that your group might be interested in. Because it's a weekly occurrence. The European Union has made that standard.
I see. That's good.
Thanks for the
That's good to hear. Thank you.
So when dealing with the amplicons from SARS-CoV-2, especially in wastewater, one of the things I saw a lot of were what we call the Lyman artifacts, which were amplicons generated from similar viruses that were definitely not SARS-CoV-2. But when you were generating your synthetic reads, were you just generating them based on the SARS-CoV-2 genome, or were you also including the possibility of amplicons from other viruses that amplified and would sort of show as a Lyman artifact?
Right, that's an excellent insight. So with for our benchmarking studies, we just solely looked at used SARS-CoV-2 genomes. But of course, that is something to consider, and also as this presentation is about SARS-CoV-2, we're living in a post-pandemic world where, of course, with surveillance, I would want to see, and where we're moving in this direction, looking at other viruses, other RNA viruses that are highly mutable as well. And that's something I would like to do for my PhD.
Thank you. Thank you for your presentation. So next we'll have the closing remarks from the man who does not need any introduction.
But my kids still use, I'll get more introduction tomorrow, you'll hear some other fun facts about Chris Mason. But we, so I want to thank all the speakers again one more time for a great day, please. again for the sponsors, and we have one last half day tomorrow. Tonight is a dinner on your own, so explore Japan, find yourself in a strange environment, chat with people who regularly give talks today. And there is, we're gonna do actually also one more group photo, because not everyone was here yesterday, so we do one more group photo and then try to splice the image together. Also, tomorrow morning is the 5:30 a.m. optional fish market. I've done it before, it's really amazing, where you can see the fish get bought and sold, and people screaming in Japanese, things I don't understand, but they sound important. And it's commerce happening in real time. It's a very fun place to go eat some stand-up sushi. And so I recommend it, if you get up early, to go there, but again, that's optional, and then we'll start eating tomorrow at 9 and finish up 12:30 in the morning. So then, real quick, we had a quick board meeting earlier, and I'll have my talk tomorrow to close up some of the summary statistics and facts about the consortium and plans. We'll also have a bit of Q&A to talk about some of the upcoming plans and priorities and ways to collaborate. And I think, yeah, I didn't have any other remarks except that everyone's doing phenomenal work, and it's great to see you all. And I think we'll see everyone early in the morning and Liz tomorrow.
5:30 in the morning, where are you going?
5:30 at the fish market, yes, if you want to go.
Just go on yourself, no organized one?
I think it's just in the, it's, we don't have a meeting point, but unless we put some details for it.
Chris, we were told by someone that it's no longer possible, that it's not. It's only starting.
It's holiday. It's holiday, one else.
You know, there's something about the
You can't come.
tourists down there.
Actually, I've not been there in 7 years or so, so I'm not, we should look online.
Somebody who lives in Tokyo.
So a few of us are going to go tomorrow by the train, and we can coordinate by WhatsApp, and we'll confirm this information for tonight.
Yeah, we'll double check, but, and if you're not on the WhatsApp group, let us know that everyone should be on that, and we're going to post a lot of information in real time about the meeting, and we'll double check. Or we can go to like a fish spot where the fish eat your dead skin off the beach, it's also interesting and unusual. Has anyone been to those? They're really bizarre, I think. The fish eat the dead skin, it's totally bananas. Maybe not, everyone's making a really gross face right now, so maybe we don't go there. That feels beautiful.
Photo?
Photo, thank you. So thank you very much, and do the photo.